Hi, my name is Bob Brown. I'm a professor of neurology here at the University of Massachusetts School of Medicine and director of the Day Neuromuscular Research Laboratory. So our laboratory is devoted to understanding the causes of and thinking about treatments for ALS. We've had a remarkably good year in no small measure due to all of the wonderful support that we've received from the Angel Fund. The focus of our work has been to understand the cause of ALS and then to use insights into the cause to think about treatment. And the particular angle that we have always taken is to look for genes whose variations or defects can cause the disease. This has been a particularly exciting year because we and collaborators have been able to describe 3 new ALS genes this year, and also to find for the first time a gene whose variations affects how long ALS runs its course. And that's, I think, very useful because it gives us new ways to think about the biology of the disease. We're trying to use this information now to develop new ALS therapies. What's also exciting is that we're on the cusp of a whole revolution in the technology for studying genes. So, for example, we're about to embark on projects in which we sequence all of the DNA molecules, and there are 3 billion of them in every cell, in ALS patients as compared to controls, to ask, are there subtle changes or nuances in DNA profiles hidden throughout the genome that set the stage for ALS? That is enormously exciting. It represents sort of the absolute gold standard in terms of ALS genetics. It's also, like these other projects, very costly to start up. It also entails high risk, but it is essentially as complete a genetic profile as one can get, sequencing every molecule of DNA. That's terrifically exciting, and we think it's going to be a model that other groups will follow as well. There's no question that the support of the Angel Fund has been absolutely indispensable in keeping us at the cutting edge of the kind of work we do. They've helped us buy state-of-the-art equipment, which allows a throughput, a volume of studies that is otherwise impossible. The Angel Fund has been critical, in particular, in allowing us to do what we would call discretionary, high-risk projects, where we think that 2 or 3 lines of inquiry are important, but we can't be sure. They fund us, we pursue them, and then if they look secure, we're able sometimes to get longer-term federal funding to keep the work going. But that initial startup, high-risk, high-gain funding is absolutely crucial to any biomedical research. And it is that, among many other roles, that the Angel Fund has done so very well for us.
So you can see this has RB and that has MS.
Okay.
So that's how you know.
The Angel Fund has funded fellows and other researchers in the Day Lab and in other labs studying ALS, which is significant because these days funding is really difficult, and so it's enabled people and researchers to get closer to finding a cure for the disease.
I wish I could visit everyone personally and shake his or her hand, because the achievements through the Angel Fund have been substantial, and they have been built upon the goodwill and the fundraising of hundreds and hundreds of specific individuals. There's no way adequately to say how grateful we are to them.